Hello, everyone, and welcome to the Data Science Zoominar today, March 23rd.
Welcome, everybody. Today, we are very fortunate to have Tim Rebbeck talking with us. He is a professor here at Dana-Farber and also at the Harvard School of Public Health. So his research focuses on the etiology and prevention of cancer with an emphasis on cancers with a genetic etiology and those that are associated with disparities in incidence or mortality by race. He's directed multiple large molecular epidemiological studies and international consortia that have been used to identify and characterize genes involved in cancer etiology. Understand the relationship of allelic variation with biochemical or physiological traits, explore interactions of inherited and somatic genomic variation with epidemiological risk factors. So today, we'll be talking about some of these more epidemiological aspects of cancer research. Thanks for joining us, Tim.
Great to be here. Thanks for inviting me.
No problem. All right, we might get questions from the audience, which I'll be asking as well. So otherwise, let me get that ready here.
Great.
So everybody, if you have questions, just put it in the Q&A section and I'll try to get to them. All right, so in research hospitals like Farber, it seems to be the case that we focus on treatment, and when it comes to the more basic science aspects of cancer research, we focus on basic biology, trying to understand cancer at the molecular level. But if I'm not correct, if I'm not mistaken, historically, the biggest improvement we've made in terms of fighting cancer and the effects of cancer on society has been through prevention. Is that right? Or is that a little exaggerated?
Well, I mean, I think it's very correct to say that the big impact in the population on reducing incidence and mortality from cancer, just in terms of raw numbers, has been through prevention and early detection. I think that's quite true. But I would also say that you can't have prevention and early detection without good treatment. And so, they all go together. And
sure, absolutely.
Yeah.
Absolutely.
Yeah. So
I guess I'm just pointing out, I'm not, let me be clear. I'm not like putting them against each other. I'm just pointing out that I think a lot of people that visit cancer research hospitals and they see what the research goes, they forget that this is also a very important part of cancer research.
Yeah, absolutely. And I guess I would say Dana-Farber and most other, or maybe all other cancer hospitals that have a strong research component are really concerned with prevention and early detection. It's true, I would say, that most don't spend the same amount of effort and energy and resources in prevention and early detection, particularly in research, as they might in basic science or drug development. I think we could always do better.
Sure, and Farber does very well, I think, compared to others. We have a whole department or a division dedicated to population studies, and that's great. We collaborate. Our department collaborates very closely with that division.
Exactly.
So much of our research has been focused on determining risk factors for prostate cancer. Can you tell us a bit about the history of the large studies that you had to organize to learn that? That's another thing that I think our audience would benefit from hearing about.
Sure.
And by the way, I forgot to mention at the beginning that our audience is mostly our department, but there's also other people that join. So it's a lot of statisticians, computational biologists, software engineers that work here at Farber and collaborate with many others. So one thing I want them to hear about is how challenging it is to perform these studies. It's because you have to get a lot of people and you have to keep track of them, and it's quite a logistics operation.
Yeah, absolutely. And I can focus on prostate cancer, but I can make some of the comments that are really generalizable to many cancers as well. I think that the evolution of the field has been that small studies were undertaken in isolated populations, hospital-based populations, that identified some early risk factors for cancer or for prostate cancer. And they were largely biased. There was a lot of study bias. There was a lot of, the methods were not optimized. And so there's a lot of noise in the system. And you'll see this because you'll hear over the decades, you'll hear reports in the news, oh, such and such is causing cancer. Such and such is associated with your risk of having or dying of cancer. And, sometimes those reports are correct, but by and large, they're not, or they're not replicated. And so people get very confused in the general population, as well as the scientific community, because there's all sorts of reports that come out, and it's very difficult to tease apart what's true and what's not. And in general, it takes incredibly large, usually prospective cohorts, meaning hundreds of thousands of people with really good measures of exposure, with longitudinal follow-up, to really tell us what's truly an effect, what's truly an association. And maybe if we get lucky, we can talk a little bit about causation, and if we add some more data to that. But I would say, of all of the data that are out there, and this is not just true for prostate cancer, the majority of things that we have been told might be associated with cancer risk or not, or we're not sure. A few things pop out as being really salient, and those are very important and maybe modifiable, maybe actionable in some way. In prostate cancer, there have been large studies, and I won't go over the list, but large, really millions of people who have taken part in large prospective studies to identify prostate or other cancer risks. And after all of that and decades worth of work and really strong science, strong, well done work, literally involving millions of men followed prospectively, we really don't have hardly any known risk factors for prostate cancer after all of that. The prostate cancer risk factors right now are age, race, particularly African American men have higher risks, family history, if you have a family history, genetics, some high risk genetics and low penetrance genes, height, maybe adult attained height, maybe, and obesity for aggressive prostate cancer. So first of all, all of the things that we've been thinking about, diet, nutrition, pesticides, exposure, smoking, lifestyle, all of those things that have been studied so much have not really panned out as being things we can actually clearly say are risk factors for prostate cancer. So, wow, that's just incredibly frustrating, because we've done good science and we still have not found a lot of things. And if you remember the list that I just gave you, almost none of those are really modifiable risk factors. I mean, maybe obesity is a modifiable risk factor, but it's not easily modifiable. And none of the others, family history, race, those are not modifiable factors. So we have a lot of more thinking to do. And just give you a caveat to that, even though we as epidemiologists can't say that there are a lot of these other risk factors, I don't think any of us think that most of them are not risk factors. We just can't detect them using the epidemiological tools we have. And so so some of them truly may be null, but trying to prove the null is very difficult. And so having knowledge about what's truly not a risk factor versus those that might have a tiny little effect on risk that we're really having a hard time defining is very difficult. And again, if it's got such a tiny amount of risk, if it's got a odds ratio, hazard ratio, risk ratio, whatever, of 1.01, do we even really care? Because you might be able to modify it, but it's going to have such a minuscule effect on an individual or a population risk, maybe it really doesn't matter. So it's been really tricky, and prostate's been worse than most, but I think you could make the same kinds of arguments for many other cancers. So, epidemiology absolutely has its limits. When you find a good one, like lung cancer and cigarette smoking, you're golden, because you can do all kinds of things. But even in that very simple situation where cigarette smoking is strongly associated with lung cancer, we see how hard it is to modify that and to make that actionable. So epidemiology can tell us a lot and teach us a lot, but it's got a lot of limitations as well.
So what about what's another, smoking is the obvious one. What are the other ones that you would list as as being discovered through observational studies?
So I think that, smoking and many cancers, not just lung cancer, but smoking and many cancers, the other kinds of exposures that I think are really prominent in reproductive factors, in many breast cancer, for example, reproductive history and hormone exposures are clearly have been defined by epidemiological kinds of associations. They're obviously also potentially modifiable. Some of them are modifiable, and have a big enough impact that we can think about how to modify risk. And that might be changing, so oral contraceptive use or hormone replacement therapy or some of these exogenous hormones. Their use has been changed by observational studies in epidemiology. Other things like endogenous, when you have your menarche or age at first full-term birth, those are much harder, obviously, or impossible to change. But yet we've learned a lot about modifying exposures or modifying risk by understanding the underlying epidemiology of some of these factors. The other big class is infection. So HPV and cervical cancer, HBV and liver cancer, H. pylori and stomach cancer, to name a few. Those have been very strongly influenced by epidemiological studies and knowledge of the effect of these specific exposures. And those have led, of course, to things like vaccination or screening or treatment of, for example, stomach cancer and ulcers. With H. pylori infection, that's, those observations have changed the way we think about ulcers and stomach cancer. So there have been quite a few. There have been also some rare-ish exposures, meaning, so for example, the Hiroshima bomb. We learned a lot about epidemiological associations between individuals who were Hiroshima and Nagasaki survivors and their cancer risks. But those are, like very rare exposures and very unusual exposures. And we have a lot of those rare and unusual exposures, but very high level exposures that taught us a lot about cancer risks. Not necessarily, again, something that will end up being modifiable in and of itself, but taught us a lot about the etiology of disease that helps us modify risk or manage risk.
Well, that's right. Radiation, we knew that from before, though, right? We knew that from like when they first started noticing a strong association between working with X-rays and getting cancer.
Yeah.
By the way, that reminds me of a question. Maybe you can.
Yeah.
I never got a good answer for this. So when you go to the dentist and they're gonna take X-rays, they'll put like something on your chest and then they'll leave the room.
Yeah. Right. Yeah.
And then they shoot your face with this thing. So what is it? How does that work? Like, why is the chest, but the chest is covered, but my face isn't? Like, what is that? Is it because the cranium is a strong enough protector of radiation?
So the issue of X-ray exposures in general, not just dental X-rays, but chest X-rays and other X-ray exposures have been studied intensively. And I think the problem is that the level of exposure that you get from a dental X-ray or a chest X-ray or things like that are fairly low. At least now, maybe.
Yeah, it's like the sun might have more. I don't know.
And you fly in planes and you get X-ray exposure, and there are also the issue is that a lot of these exposures are actually quite low today, and so the problem in understanding risk is really being able to extrapolate below what we know are measurable levels of exposure that we think cause risk. And so can you extrapolate down to a single dental X-ray or a number of dental X-rays over the course of your lifetime and add that up to get enough information about the associated risk to really say, beyond this certain threshold, you're at risk. Under a certain threshold, it really doesn't matter. And you were making up stuff a lot of the time because we don't have good data around these very low exposures. We can make the same argument about radon exposure. Extremely low levels of some of these exposures, we just don't have good measurement of those. And we don't have good studies that allow us to say these extremely low exposures are associated with a certain level of risk of whatever, cancer or whatever. So like they put the lead vest on you, I think that's just, it's fine. It doesn't hurt anything to do that. I don't know that that's necessarily based in a lot of evidence that the scatter from a dental X-ray is hitting your chest to the point that it is causing a lot of extra disease. But, and somebody can correct me if I'm wrong, but my understanding is that it's not really based in knowledge of how much an X-ray or a series of X-rays are going to affect your risk versus mitigating using things that are just easy. And even if the risk is close to zero, now it's zero. And you mitigate that risk because it's easy to put a lead shield on you and it doesn't cost much and it doesn't hurt anything, that sort of thing.
Yeah, I just want a helmet too, not just the.
Yeah, you can have the helmet, but again, do we know how much? And we do have estimates of all these things. It's not that we have no data, but the risk of, let's say, brain tumors in people that have had a lifetime of dental X-rays, if we could even measure that, or if we have a good estimate of that, it's going to be low.
Yeah.
So, is putting a lead helmet on going to make a difference? I don't think we have enough data or knowledge to say that it's worthwhile or what that would be.
Yeah, I get it. Yeah, it's just it seems so scary, especially when they leave the room and then they're shooting at your face.
And I think that there is a difference, that low-level radiation exposure, the risk is probably not zero, but it's small. Yeah. So obviously, if it was something that caused a sufficient amount of risk, they wouldn't be doing it anymore. Yeah. It would be not allowed, or would they be doing it differently? Or, the FDA or whoever it is that regulates these things would say, wait a minute, you have to stop. And so the fact that they haven't stopped tells us that there is no risk that's so huge that it's, really a problem.
But how would they know if it's a very small risk? Somebody's running studies constantly.
Yeah, I mean, by these kind of observational studies that we're talking about, very hard to do. You can imagine who gets a dental X-ray, everybody, and who gets it maybe once a year or something like that. And we need to follow people over many years, accumulating their exposure and then following them many years. If the risk is very small, this is just like a power calculation you can think about. If the risk is 1.1, which I think would be kind of high in this situation, how many people over how many years to develop certain specific cancers would you need to detect that effect? And it's huge. You would need huge studies to do that. And there are huge studies that have tried to do things like that, but it's very hard to come up with these. And again, I'm using dental X-rays as one, but you can imagine any of the exposures out there.
Microwave cell phones. Yeah, there's
all of the above. And again, people have studied this. It's not that people haven't tried and haven't done good studies, but the level of exposure and the level of association is so small that we kind of have to look at it at some point and say, it's small enough that we can't worry about it. We can't worry about all these exposures and risks that are at 1.001 or something like that. Maybe we can, maybe we decide we want to, but most of these exposures come up as null with the epidemiologic data to the degree that we can do these studies. And again, not to say that we're doing bad studies, but there are limits of detection and also limits to what we consider are regulatable, modifiable, or avoidable risks that we want to take. So again, everything that we do in life is a risk-benefit ratio. So is the risk of having a dental X-ray worth having the dental X-ray because it's going to save your teeth, because you're going to get, you're going to be able to figure out you have cavities? What's the risk-benefit ratio there? And I think the system has decided that the risk-benefit ratio for having a dental X-ray is positive, because you're going to have your teeth filled and take care of your teeth, and you're going to have, that's going to be a positive, and the risk that we take is so low that it's worth, doing. And so everything that we do is a cost benefit, a risk benefit ratio, and we need to sort of balance those with everything that we do. Okay. And that's the bottom line. Nothing that we do is without risk.
So one of our attendees is pointing out that the X-ray techs are exposed much more frequently than us. So, and we would probably see it there if it was an actual risk.
Yeah, I think that's a point I was going to raise. Glad that someone did. In general, a lot of the studies are on workers in these fields. So you can think of hair dye. Hair dye has been a source of concern about cancer risk. And so you see sometimes beauticians are at increased risk because they're being exposed so much compared to the person whose hair is getting dyed, that, you know, it's a risk benefit ratio. And if you're exposed a lot, your risk is going to be higher. And if you're exposed less, your risk is going to be lower. And the question is it a reasonable cost benefit ratio? And can you do anything to mitigate your risk in the process?
Okay, we have our first question. So this is a hard one, and you alluded to it already. Would you say that we can know for sure from observational data that an exposure has a causal effect on some type of cancer, or can we only identify associations that allow us to intervene with certainty with randomized controlled trials?
Yeah, so there are two questions there, which are both relevant. So we can know for sure from observational data if there is causation involved. So we can know that because there are criteria for causation. Again, there's a lot of data that suggests, you know, causal inference is possible from some data. Causal inferences can be made because we can eliminate the bias or the mediation effects or other factors that would help us to understand something that is truly causal. And there are methodologies that will specifically make causal inferences. But I would say that the vast majority of observational data won't result in causal inferences. They will remain associations because we can't eliminate the biases, the mediator effects, the other things that that would keep us from a causal inference. Causal inferences can be made, but they are relatively rare from observational data. And so
yeah, with smoking, we had paper after paper eliminating alternative hypotheses, and,
right.
and by the end, it seems like we can actually say it does cause. Well, it increases your risk. I mean, that's another distinction. Not everybody that smokes dies of cancer, but so right. It's really it causes a higher risk.
But I think the key is in what you just said in the second question is the reason we want to know about causation is so that we can be sure we should intervene, that we can develop interventions that are biologically motivated or whatever it might be. So I would say that, you know, do we need to know causally that smoking causes lung cancer or head and neck cancer or something like that? I would say we know enough from the epidemiological data without knowing causation or without knowing the biology of smoking-induced lung carcinogenesis that we can tell people to stop smoking. I mean, so we have enough data from observational studies to make an interventional statement and policy statements and things like that. And so the question to me really is, at what point do you have enough data, even if you can't prove causation, that you can do something that's interventionally beneficial? And that's, to me, that's the question. And sometimes knowing that something is causal is required. Sometimes it's not. I mean, I would say we can tell people to stop smoking. We don't know all the biology that underlies smoking and its effect, but we can. Do we need to tell people to stop smoking? No. So again, I think that's, you know, to the second question, I think we can get enough clear knowledge about what's going on to either make policy or behavioral changes or drive a randomized controlled trial. And I think there's a great example of the CARROT study. I don't know if you're familiar with the CARROT study. It was done 40 years ago. It was beta carotene interventions in smokers. So all the epidemiological evidence said that if you give somebody these micronutrients, beta carotene, you're likely to lower lung cancer in heavy smokers. So they did this very large, tens of thousands of person study with intervening with beta carotene on smokers. And what happened? Those in the intervention arm, those that received the beta carotene, had higher rates of lung cancer than those in the control arm. So this is exactly the point that you're getting at. We thought we had great observational data, or they did back then 40 years ago, that this micronutrient, vitamin A, beta carotene, would be enough to lower risk, and it increased risk. we didn't know enough about causation. We didn't know enough about biology then. And we went ahead and, not me, but the people that did that then went ahead and did a randomized controlled trial and made more cancer. So I think this is exactly right. The point that is being made here, how can we be sure that we've identified all confounders? What if we don't know everything that we think we know? And we would make mistakes when we don't know all about what we should be doing, even in something like a simple vitamin that should have no negative effects. It did, because we didn't know the biology, we didn't know the pathways well enough. So that was a real problem. And this happened actually in multiple studies. It happened in a study in Finland, and the study in the US, where we gave smokers similar micronutrients and increased risk of cancer in all of these different studies. We thought we knew what we were doing, but no. And so that put a huge problem for all chemoprevention studies, because they kind of thought the observational data was enough to guide these RCTs, and it clearly is not. And so chemoprevention literature really fell off because people said, oh, we can't just know a good p-value from an observational study. We really have to know what's going on causatively, pathway, biologically, all these things before we can start giving people even things that should be innocuous. And so we learned lessons, but it set the field of chemoprevention behind by decades. And even now, chemoprevention is not really where it should be, because we really clearly need to have strong biological causal inference before we want to give people anything. So yeah.
Thanks. So the person who asked that, Fernando Rutia, is also was wondering, what does it mean to be 100% sure, right? That's now getting into, that's like another session we can have on philosophy of probabilities and
right
and association. But I would say to that is that I guess technically we can never be 100% sure,
no.
but we can get practically there.
Yeah.
And it's not necessarily through one study, but through many studies and a consensus, some kind of consensus of some diverse group of scientists that get together. It could be the National Academy of Sciences or some the equivalent in Europe or some other continent. They all read all the literature and say, okay, we are convinced. And that's really, at the end of the day, how we come up, how we decide, right? It is by consensus. It's never like one with observational data.
Right.
I've never seen a case where one study just proves it, one observational study.
Yeah, and also, it's a combination of different kinds of data. So it's not just observational.
Right. Sure. Yeah.
It's the RCTs, it's the biology, it's all of the things that go into making a decision. And again, I go back to when you think about, is there a way to be 100% sure? No, I don't think you can ever be that. But you go back to the risk-benefit ratio, because everything that you do has potential risks associated with it. And, possibly. I mean, there may be things that are so safe that there are no risks, but there's always going to be some risk. Even if you give people aspirin, there's risk. Something that's common and used, and we don't need the aspirin clearly has benefits in chemoprevention for colon cancer or for other things, but there's still risk. If you give enough aspirin to some people, they will bleed. There are always things that can go wrong. So I think we really have to be realistic in terms of our risk-benefit ratio, so that the harms are absolutely minimized, but the benefits are clear, and as much data as we can collect to make that. And it's really hard when you're talking about prevention. So when you're talking about treatment, people will put up with a lot of side effects and harms because they are sick and they want to be cured. When you're talking about prevention, people's tolerance for harms is close to zero or zero maybe. And partly that depends on how much risk you have. So if you have an extremely high risk, let's say you have a BRCA1 mutation and your lifetime risk of breast cancer is 50%, 70%, and your lifetime risk of ovarian cancer is 40%, or whatever the correct numbers are, that's high enough risk that you feel like you need to do something so you won't die of this cancer. But so you're willing to take higher risks. You're willing to have your ovaries removed prophylactically, for example, which has a lot of risks and causes a lot of side effects, but that risk is high enough that you may be willing to do that. If you're doing something in the general population, those risks probably are going to be very, very low. So again, it's also the setting that you're talking about, what level of risk, what it is you're trying to prevent or mitigate, and how motivated the person is to want to put up with any side effects that might exist.
Excellent. I have one more question. Maybe I'll leave it for the end because I do want to switch gears to some of the other topics about disparities.
Yep.
So you've also done important research in disparities incidence or mortality by race. Can you tell a little bit more, like some examples, some numbers that you've discovered through these studies?
Well, a few things, few made sort of take-home points. One is that disparities exist in many ways. So we often think of disparities as by race or ethnicity. And there are some major disparities by race or ethnicity, but there are also disparities by age, by gender, by disability, by socioeconomic status, by residence. So we need to think very broadly about what the disparities or inequities that there are out there that lead to differences in your ability to get care, prevention, treatment, whatever it might be. So there's a broad range of things going on there that define disparities, and we need to think very broadly about how to understand each of those and then address them. So just about every cancer, every disease really has some aspect of disparity in it. And some are really profound. So for example, I study prostate cancer, and prostate cancer has the highest rates, highest mortality in African American men compared to essentially every other population in the world, or every other cancer in the world. It's a huge disparity, and it's incredibly complex to think about. So what does it mean to say that an African American or an African American population has higher risk than a Caucasian population or an Asian American population? It implies that there's probably some biology underlying this. There's some biology that makes the disease arise more frequently in African Americans, but it also probably reflects the fact that African Americans may have less access to early diagnosis or screening. They may get poorer care or different care. There may be behavioral and lifestyle factors that add on to this. You certainly skepticism and mistrust of the medical profession, and not wanting to have screening, not wanting to have a digital rectal exam. Those are cultural behavioral things. So suffice it to say that it's not usually a simple thing where there's one factor that's causing the disparity. The disparity is usually a complex set of things, and that you can't necessarily think about one thing at a time. Having said that, there are certainly individual actions that one might be able to take to address disparities. So for example, access to PSA screening might be a way of addressing the disparities problem, but it's only going to address one piece of a very, very complex problem. And so it might be that the disparity needs to be addressed on multiple levels. And some of those are quite fundamental, fundamental in terms of addressing structural racism in society, which is obviously not an easy thing to do. But something that might be the fundamental cause of a lot of these disparities. And so how do you think about when you want to create an intervention to address a disparity, how do you think about addressing systemic racism, which is a problem of a magnitude and depth that few individual studies could really hope to address, really. So lots of complex things going on there, but very interesting. The other thing that I would mention is that disparities are an opportunity in some ways to understand variability in disease outcomes. If you're in data science and in epidemiology, the more variation you have, the more you have to work with. If everybody was kind of the same, there would be nothing to study, right? You need variability, you need variation in order to have to study and find risk factors, to understand, what's going on to create that variability. And so disparities are an opportunity for us to understand the broadest scope of variation in data, in risk, in the population, and to use that knowledge, or to leverage that variation to try to understand underlying etiological factors. So there is opportunity in disparities. So I don't know, I can keep going, but maybe.
Yeah, no, that's interesting. So what does the data look like to answer some of these questions? Like you just gave a great explanation of how complex this can be. There's, because there's biology, behavior, and then differences in access. So those must be hard to parse out. Like, do you, in these data sets, do you have enough information to at least try to parse some of these things out?
Yeah, I mean, sometimes. And I think, the point that you're making is a really important one, because what we were talking about before and what we're talking about now in terms of how epidemiology and observational studies work is we're very focused or reliant on a p-value and statistical power and approaches that are, very limiting in a lot of ways. And I think that one of the things data science brings to us that has not been a traditional piece of epidemiology, although I think more and more it is the ability to find patterns in data and to find subsets, observations, associations that are not p-value dependent. And I think that that's been a problem that we faced a lot, is that our field, because again, we don't want to make mistakes. We don't want to have a lot of type 1 or type 2 error. We don't want to start giving people beta carotene before we know that it's really going to be beneficial to them. We are super, super conservative about what we will call an association. Mm-hmm. And again, that might be appropriate in many ways, but when you're talking about these very complex sets of factors that lead to a disparity or to a complex disease risk, we need somebody to be thinking about sort of discovery and of patterns that we can begin to build on. Because our p-values are going to miss a lot that's relevant, and we're never going to have power to find 2 or 3 or 4-way interactions among things. Data science brings that to us.
And non-linear interactions and non-linearities.
Absolutely. I mean, if you look at what we do, our traditional methods, I think the walls have been broken down a lot in recent years, but the assumptions that we make, proportional hazards assumptions, linearity, normality, all of those things are, well, first of all, we violate them all the time, I'm sure, and don't worry about it too much, but we do rely on those things, and our p-values rely on all those things to be accurate and to get the right point estimates of things. And that to me is a barrier to finding patterns. And again, I wouldn't say that if I had a neural network that found an interesting pattern, I would go right to a phase 3 clinical trial. But the complementary nature of many of the data science approaches to the epidemiological approaches are going to be something that will teach us more than if we don't cross these disciplinary boundaries.
Mm-hmm. No, and that's definitely, I can see that happening. I see a lot of statisticians getting into that world of trying to come up with new ways of looking for associations and adjusting for confounders too.
Yeah.
So that does, how much of these data sets are public? I haven't worked, a lot of my work is based on public data sets, relies on public, but I haven't really delved into the world of epidemiological big cohort studies in cancer. Is that, so I don't know, is it common to make these public, or are they kept private for further research in the future?
So the general philosophy recently, the last 10, 20 years, is that all data should be made public. And this is something that's a mandate from the NIH and other funding bodies. What hasn't happened is the data have not been made public the way genomic data have. There's no dB gap equivalent for epidemiological cohort data. And on the one hand, the data should be made public. Everybody wants to make the data as public as possible. On the other hand, there's been a lot of hesitation in the field because there's a lot of ways to misuse the data. I suppose that's true of any data set, but people are very concerned about the misuse, the misanalysis of data if you just put it all out there publicly. That's a little paternalistic and may not be really completely appropriate, but I think that people have been concerned about this. And so what usually is the case is that all of these individual cohorts have a mechanism by which you can apply for the data. And so you can say, here's my question, here's what I'm going to do as analysis. And again, it's not always a complete scientific review, but at least it's some sort of, yes, this is a reasonable question, and yes, you know what you're doing to work with these data. And they're pretty much open access after some, sort of review of the protocol. So it's not a DB-GAP where you can just go in and get things. But
so then how does, let's suppose someone in our audience wants to use that data, and is it something you email the first author, or what is it? What is the procedure?
Yeah, so again, for the large cohorts, there's a group called the Cohort Consortium, which is led by NCI, overseen by NCI. And the Cohort Consortium is essentially all the big cohorts that have ever published anything. And so you can go directly to the Cohort Consortium and apply directly to them for data from a specific study or all the studies or subset of studies. And there's a form that you can use to directly apply for those data. But you can always go to the individual PIs of the cohorts and say, I think your data is appropriate, can I get it? And they all have their own system where they, vet the protocol, the project. A lot of times it's just to make sure somebody else isn't working on exactly the same thing with the same data, but sometimes there's some scientific concerns raised, et cetera. So there's a lot of different ways of accessing it, but it's always, these days, everything more or less is intended to be available to anyone who's qualified to use it, I think.
Okay, that's good to hear.
Yeah.
You should call it the cohort of cohorts, that group.
Yeah, it is kind of, that's what the Cohort Consortium is, because they figure out how to harmonize the data among all these cohorts and bring them together. So yeah, it is essentially that.
Good. So the last, we're running out of time. We ran out of time, but I do want to ask this last question. So there's a lot of, you alluded to this earlier, but there's a lot of news media articles that have a headline saying X causes cancer, and they come out all the time, and often they come from one study on animals or some very small observational study. It's hard to tell from the news article. So what do you recommend we do, those of us that we see an article like that, what would you do to just find out what exactly is this, like, because often they don't even have the paper they're talking about.
Yeah.
So is there maybe a place where you have like a list of everything that's come out that's a big core that has been done that has had some kind of consensus about?
Yep.
Like, how do you parse out all those?
Yeah. Well, as you can imagine, that's really hard because the science is not always clear, even after you've gotten a good study. And ultimately, it's very confusing to the population because we keep changing our mind about things. On the other hand, it's actually what's supposed to happen. We're supposed to publish science, and the science is supposed to change as we gain more knowledge. So it's a good thing that new science is coming out. Your question about how we decide what's real or not, there are a number of locations, the NCI, the CDC, IARC, a number of places where we have good information about what's real and what's not. Oftentimes those come from systematic review and meta-analyses. Mm-hmm. Things that are really very clear often have had a meta-analysis that sort of says, yes, we believe this, it's been done enough. There's consistency, there are common point estimates, there's enough knowledge that's aggregated that we can trust this result. And so many of the major websites, CDC, NCI, et cetera, summarize what we know AICR, the American Institute for Cancer Research, does that for nutrition. So there are sites that are sort of trusted sources that have summarized the knowledge, summarized the systematic reviews and things like that. And there are also sort of Snopes-like websites that are like, this is false, this is not real. Antiperspirants cause breast cancer, things like that.
But let me say, there's something in between. That's true.
Yeah.
Those are easy to catch.
Yeah.
But there's some that are in between.
Yeah.
It's like the one study came out.
Yes.
And it's a good study, and they did it right, but it's just one,
yeah.
as opposed to a consensus of many papers. That's the distinction that I think is a little harder to make for us, for those that are just reading the regular press.
And true for us in the field as well. I mean, one good study is provocative and interesting. It makes you want to follow up more. I would say if that study is not, you can do and we do all the time, a formal grading analysis of, what is this free from bias? Is the study got a sufficient power? Is it, there are objective ways that we look at data and results to make sure that we get a strong grade for the study, and that helps to do it. But with one study, it's very unlikely that that's going to translate into something that is an intervention that people will use, that will be a policy change, a practice change, et cetera. And so I would say one good study is interesting. Very rarely is that a policy or practice changing event, particularly in the observational epidemiological literature. I wouldn't say there isn't any ever, but I would say that would be unusual, and we wouldn't take a single good study as to move forward with in any kind of a public health or practice kind of way before we have that aggregate data validation, things like that. And then also, most of these studies are done in white majority populations. Translating that out into minority populations or other groups is always, how generalizable this is, things like that. There are a lot of other questions that fall down to whether you can make this a practice or policy changing event. And so it happens very slowly, and it's very difficult for all of us to try to get to what's a good enough amount of data with quality to believe it in a policy, whatever setting.
Great. All right. This has been very instructional, interesting. So I want to thank you for participating, for answering all our questions. If you have any last words for a group of statisticians, computational biologists, other data scientists that might be interested in this area, are interested in helping in any way or getting involved.
Just to say that we need you, the people who are thinking creatively about analytical tools or design tools or whatever. The fields of epidemiology and data science shouldn't be distinct as they are. We should be able to talk to one another more, and there's a lot that can be done if the novel methods and thought approaches in your field can leverage the things that we're trying to achieve in epidemiology. So please contact me or anybody in the field to try to work together, build methods, because we really can do more together.
Excellent. Well, thanks again. Bye-bye, everybody.
Thank you. Take care.
See you next time. Bye-bye.